Hi everyone, welcome to the Interagency Modeling and Analysis Group Multiscale Modeling webinar series, and we are pleased to have a presentation by Dr. Thomas Yankeelov. Dr. Yankeelov is an Ingram Professor of Cancer Research and a Professor of Radiology and Radiological Sciences, Physics and Astronomy, Biomedical Engineering, and Cancer Biology. He serves as Director of Cancer Imaging Research for both the Vanderbilt University Institute of Imaging Science and the Vanderbilt Ingram Cancer Center, where he's also the co-leader of the Host Tumor Interactions Research Program there. He received his Master's in Applied Mathematics and a Master's in Physics from Indiana University, and his doctorate in biomedical engineering from SUNY at Stony Brook, where he completed his dissertation at Brookhaven National Laboratory in 2003. His research program focuses on the development and application of in vivo imaging methods for predicting treatment response in cancer, and extends from mathematical modeling to applications in preclinical models to implementation in human studies. And we are very pleased to have Tom present this presentation, which comes from his Multiscale Modeling UO1 Award from the National Cancer Institute. His UO1 Award is titled Image-Driven Multiscale Modeling to Predict Treatment Response in Breast Cancer. And we are very pleased to hear your presentation today. So Tom, you're welcome to take it away.
Okay. Thanks so much for that introduction. I'm hopeful that this will be an interactive discussion, so please feel free to interrupt me at any time. Also, in trying to communicate that this is truly a multidisciplinary team effort, there's going to be two other speakers who pop in at various times. Darren Tyson will pop in towards the end, and then Jared Weiss, who works very closely with Mike Miga, will pop in the middle. Can you see me move the arrow? Can you see the cursor move on the screen?
Yes.
Yes. Great. Okay. Now I cannot. All right. Very good. There it is. All right. So I'll start by trying to motivate the problem to you. So when a patient is diagnosed with a solid tumor malignancy, they will come in, and their standard of care, the standard of care for this is to get a series of high-resolution CT or MRI scans, as in the example here. So this is an axial cross-section of the CT. Here's the vertebral column right here, and this is the liver right out here, and there's two lesions titled tumor 1 and tumor 2. And so what happens is that the longest dimension of these lesions are written down. That's what this is right here, in millimeters. So tumor 1 is about 24 millimeters across, and tumor 2 is 14. And then these are summed. This is the sum of the longest dimensions of the tumors, and this number is written down right here. So it's about 3.8 centimeters. And then the patient will go on therapy, and they will come in and they will have various checkups at various times to get follow-up scans. And so here's at visit 2, here's these two lesions as well. There's been about a 5% increase. By visit 3, there's a 16% increase. Visit 4, it's now 32%. And visit 5, it's nearly a 90% increase. And so a major problem with this, obviously, is that this therapy is not working for this patient, but it's very difficult to determine that just from these early time point data. In fact, if you look at the spread in time between these, you can see that it's actually quite a problem. So this is 2 months time between baseline and visit 2, a 5% increase, which is probably within just the noise of making these measurements. After 4 months, there's a 16% change. After 24 weeks, so 6 months, there's now a 32% change. And this is when we would start to now clinically say that this is progressive disease. And so how do we call this progressive disease? This is all based on something called the Response Evaluation Criteria in Solid Tumors, or RECIST. It's now about 15 years old, and the idea was to come up with a simple way to assess therapy response in tumors to treatment. Sorry, I left off the word treatment over here, but that's what it is. So it's divided up into 4 categories. So once again, the patient comes in at the beginning, gets that high-resolution CT or MRI. The longest dimension of the tumors is summed, and then at the conclusion of therapy or at midpoints, they're also summed up, and you compare them. So obviously, if the tumor completely go away, that's a complete responder. If there's a 30% decrease in that number, the sum of the longest dimensions, it's a partial response. If it gets bigger by 20%, then it's or the appearance of new lesions, then it's progressive disease. And if it's none of those three, it's stable disease. So in that example on the previous slide, we weren't able to say that that was progressive disease until after 6 months, after 24 weeks. So we need to be able to figure out very early in the course of therapy when patients are responding or when they're not responding, so that we can move them to other therapies which will potentially be more effective, plus eliminating toxic side effects that many of these therapies have. The problem with the RECIST criteria, and this is certainly not my own, this is well recognized by the field, is that it's all based on anatomical changes which are limited and non-specific and often occur long after any underlying changes are apparent at the physiological, molecular, or cellular level. And so there's my first hint at the multi-scale component of it, is that we're going to be trying to look at the physiological, cellular, and molecular measures of tumor response. So we need more sensitive, reliable methods for predicting response, and we need to do this early in the course of therapy. So the overall hypothesis of our group, and we wrote up in this perspective here, recent, I guess maybe last year, the idea was that readily available multi-scale imaging techniques can provide the data to initialize and constrain predictive models of tumor growth and treatment response, and you can do this in the clinic. So this is how I see the next approximately 40 minutes of your life going. I'm going to give you a quick overview of dynamic contrast enhanced MRI and diffusion weighted MRI, and I'm going to show how we can use this to initialize a reaction diffusion model, and then we'll extend that a little bit. That's when my colleague Gerald Weiss will come in. And then we'll talk a little bit about then extending this model forward. Include PET data, and then then Darren Tyson's going to step in and tell us about how we're making inroads into making measurements at the cell scale to then link to the physiological and sort of mechanical scale. So I hope everybody is suitably motivated. I'm on I can't see your expressions right now because I'm in full screen presenter mode, but I'm trusting that you're very excited. Is that the case? Everyone's muted too. All right, awesome. All right, here we go. All right, so I have to give one slide on dynamic contrast enhanced MRI, and it's actually a very simple technique and principle. You inject a contrast agent into the plasma while a patient is in the MR scanner. You take a series of images while the contrast agent is coming in and then exchanging between the plasma and the tissue space. And if you've ever seen compartmental models before, then you know that this is just a simple first-order ordinary differential equation, and I don't know why the derivative is not showing right here, but this should be d by dt of the concentration in the tissue is equal to the change, or is equal to the rate at which it's coming in minus the rate at which it's leaving, and the solution is just this simple integral equation here. So the idea is that if you can measure this term right here, the concentration in the tissue and the concentration in the plasma, the C sub p term, then you can fit that to a model and get out these parameters that relate to the transfer and how it leaves again, and the tissue plasma volume fraction. That's this piece over here. So here's an example of this. This is an axial cross-section of a mouse with a mammary tumor right over here. And as you take these baseline images over here, the contrast agent is injected about here, and then different tissues have different enhancement characteristics, which you then analyze with this model right up here. And so you get a different value of a parameter, and this is a K-trans map. So this is the delivery, the perfusion permeability index of the vessels delivering the contrast agent. So each different voxel gets a different has a different set of parameters, and you can make these maps to look at tumor heterogeneity. Here's an example of it in the clinic. This is pre-therapy, after at baseline before therapy begins, and this is after one cycle of chemotherapy. This is in the neoadjuvant setting in breast cancer. So here's someone who has a response. You can see the number of red voxels goes down, so indicating less blood flow, less permeability. As a non-responder, you actually see an increase in this, and so you might predict that this person's going to be a non-responder, and at surgery, that was the case. So we've done this now to a number of patients. I think it's now up to 39 or 40, and when you do the ROC analysis, receiver operator characteristic analysis, to see how good you are at predicting response, you get about, in some sense, about 77% of the time you get the answer correct. So I'm showing you this because it's a technique that's readily applicable to the clinic. People are using it to predict response. The numbers for accuracy are not not enough to change the standard of care, but they're going in the right direction. So that's dynamic contrast enhanced MRI, and I want to tell you briefly in the same manner about diffusion weighted MRI. And this is all based on Brownian motion, one of Einstein's papers in 1905. And the idea is that in free solution, after some time t, water molecules will travel some distance L from where they started. In tissue, though, there's compartment effects that hinder movement, so you get restricted diffusion. And here's a seminal paper that shows this. This is with packed cells, and as the volume fraction of cells increases, the apparent diffusion coefficient goes down. And so there's, at least in well-controlled situations, there's this nice linear relationship between cell number, cell density, and this apparent diffusion coefficient. So here's a picture of this. The idea is that if you've got if you have a very cellular tumor here, and you then get a distribution of apparent diffusion coefficient values, where, which are indicated by this distribution here, and it's centered around a small number. But if you give cytotoxic therapy over time, the cells will lyse, they'll fall apart, and then you might get areas of necrosis, in which case the distribution might broaden and then move to larger values. And if this distribution is significantly different than this one, it's a marker of treatment response. And so people have also done this in the clinic. And here's a similar study. This is, again, breast cancer. This is a sagittal cross-section. Here's before therapy, and after one cycle of therapy, a responder shows an increase in the ADC, a non-responder shows a decrease. So the decrease means that there's more cells, more barriers, water cannot diffuse as far per unit time, and that's the opposite of in the case where there's a responder, where the cells are dying, there's more space for water to diffuse freely, and the ADC goes up. So again, doing the ROC analysis, you get these values, which are increasing a little bit better than the previous example with the DCE MRI. But then we can combine them, right? So this is one patient, pre-therapy and after the first cycle of therapy. And this is the DCE right here, and so this is going down, and so you think this is an indicator of response, and this is going up, so this is also an indicator of response for the ADC. Less cells, and less perfusion, so that's good. And that patient did achieve a complete pathological complete response, so there was no tumor present at the time of surgery. Whereas a patient with residual disease had this increase in K-trans and a decrease in the ADC, and so you might hypothesize that that's going to be a non-responder, and that's in fact what we found, that there's tumor at the time of surgery. And so when you compare the RECIST, which is what we started with, saying the longest dimension, the area under the curve is not very good at all. 0.5 is guessing. If you go and look at the volume, it's basically the same. But if you combine these two measures, you're getting close to 0.9, which is really where you're going to start turning some heads. So this is an indication that these quantitative imaging techniques are predictive on their own in a statistical sense, and so maybe they're appropriate for incorporating into models, which brings us to our first bit of modeling. But now might be a good time to stop and see if there's any questions about those two techniques, because I'll be referring to them a lot in the next two components. Are there any questions or thoughts or cries of joy from the audience? Just smiles and happiness? All right, sounds good. My gnome is very excited so far, so I hope you guys are as well.
All sounds good, Tom.
All right, thanks. All right. All right, so this is the reaction diffusion equation. You've probably seen this many times. If you haven't, just real quickly, the left-hand side is the rate of change of the number of tumor cells in position X and time T. And using imaging data, this is really nice for us because we're naturally gridded on the imaging voxel. A voxel is just the contraction of the word volume element, and so we're naturally gridded on that, and that's what our positions are. So then we have the diffusion term right here, right? To spatial derivatives is a diffusion term, and here's our diffusion of tumor cells. And this diffusion is different than water diffusion, which I just finished talking about. The water diffusion lets us get cell density, which is the term N sub Tc, right? That's tumor cell number. Whereas this dTc is whereas this is diffusion of the tumor cells themselves. So the diffusion term, and then here's the reaction part. This is the growth term, and here we've selected logistic growth, which is just growing up to a carrying capacity theta. All right, so we're going to run some simulations and some in vivo tests here. The basic idea is that we're going to fit this model to serial DCE and diffusion data to extract parameter values. The parameters that we want to get at are this proliferation and this diffusion term right here. Run the model forward in time with those two parameters, compare it to both in silico data and in vivo data. So we're going to do that. That's what this says. We're going to use simulated data to test the stability of the approach and see if it actually works. And then we're going to use experimental data to test the biological validity of the model. So here's a simulated tumor data set. Here's a cross-section of a rat brain that's been extracted from an MR image set. This is the corpus callosum right here to orient you. And this is a real measurement of the ADC of a tumor in a rat. And so we take this and project it forward in time using the reaction diffusion equation, and we get this simulation. So then we select a subset of the early time points with the reaction diffusion equation and minimize the area between the tumor described by these estimated parameters. We'll get a diffusion term for water and white matter, and in gray matter, that's what the W and the G stands, and then a spatial map of proliferation. So we take early time point data, in this case days 1 and 3, fit it to the reaction diffusion equation to get this set of parameters here, and then use that to run it forward in time and to predict what the tumor looks like in the future, and then compare it at the region of interest and then at the voxel level. So just to be clear, because this is the reoccurring theme for the rest of it, we have a whole series of imaging time points. We take the first 2 or 3, fit that to the model to extract patient-specific or rat-specific data, if you will, get those parameters, and then use that in conjunction with the reaction diffusion equation to run it forward and make predictions that we can then compare to experiment. So here is the simulation results right here. This is true, meaning this we assign parameters, and this is what we grew for going forward. This is predicted growth using the first 2 time points. And you can see it matches pretty well. If we use the first 3 time points, it matches a little bit better. So this is just in simulation. So here we know, so if we quantify that, then here's percent error in tumor volume. This is the dice similarity coefficient, and this is the concordance correlation coefficient. And so this tells you if you get the tumor size correct. This tells you, the dice tells you if you get the placement of the tumor size correct. And then the concordance correlation coefficient tells you about intratumor heterogeneity. Are you getting things right at the voxel level? Whereas these first 2 just get things right at the region of interest level. So if we use 2 time points, we get these values, that's the orange. If we use the first 3 time points, we get the blue. So it doesn't really matter which one we pick, but if we pick the blue one just because the results are better, then we stay within about 6 or 8% error in the tumor volume, projecting 6 days forward in time. And so 6 days may not seem like a lot, but from the time we start the experiment to when they finish, it's only about 11 days before the rat has to be sacrificed. You inject the tumor, it takes about 10 to 12 days to form, then we put them on study, but then about 10 days after that, usually about 21 days total after injection, that's when the animal has to be sacrificed. So we can accurately estimate D and K in the presence of experimental noise, and then when we project forward in time, these are the errors we get. So we do really well for tumor volume, we do really well on placement of that tumor volume, and then even at the voxel level, we're still up around 80% 6 days forward in time. Now again, remember, this is all in simulation to test the validity of the approach and if the model is stable to what you might find in the experiment. All right, so we then take these, this approach and go in vivo. All right, so here is, where's my cursor? There it is. All right, so here's day 1. So this is a real tumor that we measured. This is, again, a rat brain. This is going forward up through day 10. So this is really right at the end of when you have to kind of sacrifice the animal. So if we just use the first 2 time points to get the tumor diffusion numbers and then the proliferation values and run it forward, we don't do very well at all. If we use 3 days, we start to characterize the growth shape of it, but this area right here of necrosis, right there in the middle of the real tumor, is not picked up at all in this model. If you squint, you can maybe convince yourself it's starting to form right here, but that it's not really there. So then, not surprisingly, when we look at the percent error and the tumor predicted tumor volume, how it overlies with the real data, and then the ability to look at the voxel data, it's all quite poor. So we get greater than 25% error in the tumor volume predictions, and the concordance correlation coefficients are less than 0.3. So the conclusion here is the reaction diffusion equation is purely an incomplete description of tumor biology, at least within this tumor model, the C6 rat model of glioma. And the reason we did this was because the reaction diffusion equation is used an awful lot in trying to describe how tumors grow, and then extensions thereof of how they respond to treatment. So we were not aware of anybody who's actually tried to use in vivo imaging data to lock down the parameters on a rat or patient-specific case and then run it forward to see how it does with reality. So we feel like the approach is numerically stable and it's legitimate, but when you try to apply it to in vivo data, then it doesn't capture all the biology. So we've been progressively trying to extend this model, and I'm going to hand it over to Jared now for his discussion of how we're including mechanical properties of the tumor. So.
All right, thanks, Tom. As Tom said, kind of start with the same image-based reaction diffusion modeling approach, and then here the extension that we're looking forward to is really towards understanding some kind of biophysical concepts of the surrounding tissue matrix, specifically in a mechanical sense. So really the motivation behind this work lies in some supportive literature that suggests that there's a pretty strong empirical evidence between the tissue structural architecture and how tumors actually grow and respond in space. And then so the kind of the work that we've done to extend the reaction diffusion model to incorporate this sensitivity to surrounding tissue matrix is through this mechanical coupling. So in this top equation, which is just the reaction diffusion model that Tom just presented, basically once again just describes the sum of random cell diffusion and logistic growth. And then we extend it to include the sensitivity towards surrounding tissue matrix stiffness through the middle equation, the mechanical coupling equation. And really the mechanical coupling here we're describing as a mechano-inhibitory effect of the surrounding tissue stiffness really through this diffusion term. So the empirical evidence supports the fact that as the tumor grows into space and into areas of higher accumulated solid stress, that it really serves to inhibit the diffusion of that tumor cells. So we're representing that here by this exponential decay term where an effective tumor cell diffusion is being modified by the von Mises stress here, just being described as a descriptor of the overall stress state of the tissue itself. And then the bottom equation, we kind of wrap it all up with a mechanical equilibrium statement, which really just governs the tissue displacement due to a expansive force that's really caused by that tumor growth. So this is commonly thought of as a mass effect. So as the tumor's growing into space, that it pushes on surrounding boundaries and structures, and it serves to increase a solid stress state. So, you know, really combining these three equations just kind of qualitatively, if a tumor is growing into space, it's going to start pushing onto surrounding structures, and depending on the stiffness of those surrounding structures, we're going to get increase in the accumulated stress, and that's really going to govern how the tumor is going to grow and respond and serve to kind of shape the direction of motion or response. So really that's just kind of based on a simplistic kind of incorporation of you know, another biophysical effect that we can observe through image-based acquisitions. So then really kind of moving forward to some of the data and some of our early approaches with this, we can see if we back to this breast cancer response system here in the neoadjuvant setting. So we see here we have a time 1, which is a baseline prior to going on neoadjuvant chemotherapy. And then so if we acquire some MRI data of just the structural anatomical images, along with the apparent diffusion coefficient data that Tom mentioned earlier to give us estimates of cellularity, in this kind of top row of the time 1, we can populate a tumor distribution of cellularity. And then we can make this similar measurements on the same patient, do some longitudinal co-registration at a different time point, which is after the initiation of chemotherapy here, designated as time 2. And here time 2 is really after about 1 cycle of neoadjuvant chemotherapy, which represents 1 week. So once again, we can do this image acquisition here, get some structural anatomical information here, and then also populate that imaging domain with estimates of tumor cellularity. And then so it's represented here in this lower right image. And then looking between these 2 time points, this time 1 baseline and this post-cycle, post-1 cycle time point, we can use our mechanics coupled reaction diffusion model to really simulate what's going on in between these two time points. And we can run this model forward in time based on data from time 1 to moving towards time 2, and put it within a iterative optimization to kind of really reconstruct the driving parameters that force the model. So here we're looking at proliferation and diffusion of tumor cells. So into a little bit some of the results, we see a proliferation, estimated proliferation map done through iterative optimization here in the lower left of K, which represents that proliferation. And here it's important to note that this model really works in forward or reverse, and in forward with a positive proliferation represents growth, and then a negative proliferation here represents death or response to this therapy. So based on this kind of simulated, estimated parameter map that we can reconstruct from these early imaging time points, and then also based on the measured time 2, we can use the same model again forward in time in order to predict the cell distribution at some future time point designated as time 3. And here time 3 is really towards at the end of the patient's treatment cycle. Here in this system, at the end of neoadjuvant chemotherapy is designated by approximately 8 to 12 weeks after the initiation. So we're really using this very early imaging time points at baseline and after one cycle, along with this relatively simple macro scale model in order to predict patient response at the end of therapy after those 8 to 12 weeks. And we see here on the top right figure here, we see a simulated cellularity using the mechanics couple model. And then obviously if we remove the mechanical coupling, we just have a standard reaction fusion model, which was presented in the earlier slides here on the lower image. And if we can also compare this to actual acquisitions at experimental observations at time 3 of this patient. And this patient was designated a responsive patient. So you see here there's no residual tumor burden at this conclusion time point. And we see here this mechanics couple model really pretty accurately predicts this response with no residual tumor burden, whereas this a non-mechanics couple model, standard reaction fusion model, leaves a bit of residual tumor burden here. So then we can also do this, with other patients. So here is a different patient. So same concept of doing an iterative iterative estimation of the parameter map. Lower left here we see the proliferation map, and then same thing using at the time 2, we can drive this model forward in time, predict the patient tumor status at the conclusion of therapy. And we see here we're predicting residual tumor burden with the mechanics coupled model as a more focal tumor, and then residual tumor burden without a mechanics coupled model as this kind of relatively diffuse disease with several spots. And then comparing that to actual observed data, we see a pretty good capturing of the effects of what's actually going on in this patient with the mechanics coupled model that more accurately characterizes the spatial distribution compared to the non-mechanics coupled model. And then, so if we kind of look at this on a overall scheme on patients exhibiting a broad degree of responsiveness to therapy, we can designate patients that are responders that have no residual tumor burden at follow-up, and patients that are non-responders that have some degree of residual tumor burden at follow-up. Look at our cell number predictions seen here. And we can do a similar ROC curve in order to look at our predictive performance of the mechanics couple model. We can see here in this lower table, as observed by area under the curve, we see a much enhanced area under the curve using the mechanics coupled model versus the standard reaction fusion model. And with that, I'm going to throw it back to Tom for the rest of it.
Jacob, did you want to ask your question that you wrote on the group chat?
Yeah, you're showing us some images, and those were taken within some time between each other. So there are several issues with accuracy of the images themselves. So probably you have some registration issue, and of course you have resolution issues. And basically, how accurate is this? Did you assess your accuracy of those techniques using simulation or some other ways?
I think you're asking a question about the accuracy of the registration between scans that are separated in time by a week or so. Is that what you're asking?
You're showing us some identification. You run some algorithm to figure out where the tumors are. But each time you take a different image, the patient will be either moving sometimes, or sometimes whatever body part will be positioned a bit differently maybe than usual, or different orientation. Those are errors that accumulate. There are several sources of errors here. And you're saying something about accuracy. So first of all, the material in which you're working, the images themselves have some inaccuracies, and the registration algorithms. But by the time it comes to the point that you actually are running your algorithms, it already has some inaccuracies. Can you assess how much is there? Did you run some models to assess this?
Yeah, that's a good question. And we spent a very long time trying to work out the proper way to do longitudinal registration of images of the breast that are separated in time by by a week or more, because the breast is not like the brain. It's deformable. There's not as many internal fiducials as in the brain, where a lot of the registration work sort of grew up and was invented. So there's really not a very large literature on that. And so we spent a very long time trying to sort that out, and healthy volunteers and phantoms and in simulations. And we wrote we wrote 2 papers on it. I'd be happy to share them with you, that tried to introduce the technique and then sorted out the errors inherent in it through both volunteers and in simulations. And the bottom line is that we feel like we can get within a voxel error, so an individual in the voxel in a tumor over time after the longitudinal registration. It's a constrained longitudinal registration scheme. After we apply that, we feel like the error is within a voxel. And a voxel might be a couple hundred microns in the in-plane resolution, and in through-plane it can be a couple of thousand microns. So that's the scale of the error.
Thank you.
Sure. Yeah. Thanks for noticing that, because we spent a lot of time on it, and it's a very subtle problem, and it sort of never gets brought up. So thanks.
Yeah, I agree.
We have another question from Alpay.
Yes, hi, Alpay Özcan from Virginia Tech. So far in the talk, you have spoken about DCE and ADC, so diffusion and DCE MRI. Maybe I'm missing something, but my understanding is that within the model, you use the ADC to predict or to estimate the number of cells. Has DCE been touched, or has it been used yet, or did I miss something?
It has been used. We didn't touch on it because so far the way it's been used and what we presented has been sort of boring. It'll have a sexier entrance later on in the presentation, but so far we've used it in 2 ways. One is that since at least in the breast, and also actually in the breast, in the brain, in the breast, adipose tissue is very poorly vascularized. So when there is a tumor, when you inject contrast, it really lights up. So the DCE first helps you identify where the tumor is.
I'm sorry. Maybe I posed the question badly. Within the model you showed, have you used, or within this presentation up to this point, with the model you showed, was DCE used within the model? That's what I was with the diffusion reaction model that you were showing.
It's been used in 2 ways. One was first to identify where the tumor was, and then we went, after identification of the tumor, then we went in and sorted out through the diffusion data what the tumor cell number was on a voxel basis. It was also, the DCE data was also used in the formulation of the carrying capacity theta that's in there. From the DCE data, you get information on tissue volume fractions. So given a voxel, how much of that is plasma space, how much of that is extravascular space, and how much of that is extravascular intracellular space. And those fractions contribute to what the carrying capacity of the tissue can be. So it's been used in those 2 ways. Later, when we incorporate treatment, which I think might actually be the next set of slides, we'll incorporate a DCE more explicitly, I guess.
Okay, but it doesn't appear. The only appearance in the equation is n, the number of cells, nothing else.
Well, it appears in theta, the carrying capacity there.
Okay.
That's logistic.
Okay. Okay. Sorry. Thank you.
Yep. All right. So now, hopefully it's been clear. So first, what we did was tried to enter. The whole thing is trying to do modeling in the clinic with patient-specific data. So first, we introduced two techniques, DCE and Diffusion, that you can get good data on a patient in an actual clinical trial who has cancer and so forth. So then we talked about using that first for the reaction-diffusion equation. And in vivo data and in silico, we showed that the reaction-diffusion equation on its own isn't very good. Jared talked about using coupling that to the mechanical properties of the surrounding tissue, and we do a little bit better. And so now we're going to try to incorporate some other imaging data that we can get our hands on. All right, so we'll go back to the reaction-diffusion equation. So here's the rate of change of the number of tumor cells. Here's the diffusion. Here's the logistic growth. Now we're going to have some chemotaxis, which is going to be in the direction of glucose, up the glucose concentration gradient. And then now we're also going to introduce death. This is going to be coupled to a PDE for glucose, the change of glucose as a function of time, which also has a diffusion term, a delivery term, the way it's being used at the site, and the way that it's being removed at the site. So these are all obviously sort of conservation of stuff, conservation of mass equations, if you will. So the rate of change of glucose is due to how it's moving around inside the voxel and to neighboring voxels, how it's delivered due to blood flow, which is where the DCE can enter in, and then how it's being consumed locally by the tumor cells, and then how it's being removed by going back into the vessel and on its way. So, so I think, yeah, so here's the type of data we can get. We're first working in the brain, and I'll bring this back to the breast data in a moment. But here's the anatomical T2-weighted MRI. This is the brain that's right here. There's the corpus callosum. There's the tongue. The big jaw muscles of the rat are over here. So this is our high-resolution image that we use to longitudinally register from time point 1 to time point n. Here's the diffusion data of the tumor right here. So there's a lot of tumor cells here at the end and less so in the middle. Here's the dynamic contrast enhanced data, which shows you the proliferating rim right here and not so much in the middle. And then here's an FDG PET scan. If you're not familiar with FDG PET, FDG stands for fluorodeoxyglucose. It's a glucose analog, and it's used in the overwhelming majority of all clinical cancer PET studies. I'd say it's easily over 90% of those studies. And so the ideas are at least the way our data is acquired here is we get MRI on these days and PET on these days. And then we try to project forward in time. So these are days post-tumor implantation. So day 9, day 10, this is really when the tumor starts to be 200, 250 cubic millimeters in size. And then by day 21, really, it's sort of all over, and that rat has to be sacrificed. So I actually have a movie here. I hope this will play. So those are the data that we use to populate the model on the previous slide. It's sort of the same idea. We have more measurements. We're trying to fit for, in this case, rat-specific parameters and then run them forward in time. And so here is these are slices through the rat brain. So this is the rat brain volume, and then over time you see the tumor growing, and it gives rise to this necrotic core right here in the center. And I think this just goes for 17 or 18 days or something like that. Yeah, 17 days. And so this is what we would predict our tumor to be. So if we pull out individual times, which hopefully it'll pop up right here. So this is what we simulate the tumor cell number to be from days 9 to 17. And so this is our prediction. And all of you right now are on the edge of your seat because you know the next column is going to be the actual experimental data, and you want to see how well it matches. And the answer is we sort of do okay. We don't do great. Here we have the general shape is matching pretty good, and we get this presentation of a necrotic core, which is sort of there as well. It's not spatially aligned, but it's not awful. If we pull out the data right here and try to show you what the sort of quantify these pictures, then the simulated data doesn't sort of overshoots it and then catches back up at the end. So that's where we are in trying to incorporate logistic growth. The reaction diffusion equation with mechanics coupled and throwing in a diffusion, and throwing in glucose metabolism. And we're excited about this approach because we have an ongoing study in the breast. This is in the neoadjuvant setting. And so we're getting all of these data in patients as well. So if we can sort out how to do this properly in the well-controlled setting of the rat brain tumor model, then we might have some hope for applying it in the breast cancer case. So these are sagittal cross sections of a woman with an invasive ductal carcinoma. They're on neoadjuvant therapy, and I should have said this at the beginning. The neoadjuvant setting, if you're not familiar with it, is a really nice place to work with predictive modeling because there's a relatively short window between diagnosis and surgery. And of course, at surgery, you get the right answer. You get to know whether or not the tumor bed is there or if it's all fibrosis and the tumor's gone. And that time period, depending on the therapeutic regimen and various subcategories, can be between 8 and up to about 16 weeks. So we take images at the early part of that 8 to 16 week window, and then we have the data from surgery to be able to test whether or not our prediction is accurate. So we feel like this approach is eminently testable or translatable. So now we've talked about things sort of at the physiological and sort of bulk scale level and coupling it to mechanical properties. And now my colleague Darren Tyson is going to come in and tell us how we've been trying to link this to the cell scale, which is still a work in progress, the linking part.
Thanks, Tom. So our work primarily is, as Tom mentioned, on the cellular scale, and all done on cells in culture. It's a very data-driven, so our approach is using time-lapse fluorescence microscopy of individual cells, many individual cells over time. And we use a couple of fluorescent reporters to label the cells, one for all nuclei and one for cell cycle position to indicate whether a cell is progressing past the G1S transition into mitosis. I think you can see a little movie of the cells demonstrating a mitotic event and changing color. So they start out red and they turn yellow as they pass the G1S transition, and then they divide and go back to red again. So from this information, so from all of these digital images, we track a number of individual cells over time and assess their behavior. Our three main behaviors that we are quantifying are cell death, division, and entry into quiescence. And from that, we build our predictive models. So the single cell measurements over time, these cell fates being measured over time, are then converted into rates based on our quantitative assessment of those behaviors over time. And those rates then get plugged into a ordinary differential equation two-compartment model where there is a dividing cell population and a quiescent cell population. And there is a transition from the dividing cell population into the quiescent population with rate Q. There is a replenishment of the dividing cell population with rate P, and then the death out of both of those compartments with rate D. And these terms are, we're trying to integrate them directly into Tom's equation with the NT, and total number of cells. So trying to take apart the different cell fates that could be giving rise to the change in cell number within each voxel. So to put this all into perspective, our goal is to essentially assess the cellular behavior that could be attributed to the changes in cell number that is being detected in the patients in each voxel, which we've estimated to be about 50,000 cells. Is that about right? So anyway, the assessment that we do gives us a time-dependent prediction of the cell fates progress over time, so that the fraction of each of the cell fates within the population that these cells have achieved over time. And the graphs on the bottom are essentially our assessment of the different rates that go into what would apparently be exponential growth. So what is shown with the circles are the actual data, the change in cell number over time in a log 2 scale, which is represented as population doublings. And then the rates shown in P, Q, and D are our actual calculated values of the rates from single cell measurements. So predictions, the lines that are projected on there, are actually the predicted population level changes based on the single cell fates, the rates of the single cell fates. And you can see if only, so this dashed line in the DMSO, the non-drug treated condition, the control treated condition, would be if division only were taking place. And with that alone, it essentially is overestimating the apparent growth of the population. So there is, even in an exponentially growing culture in vitro, these different rates are actually being achieved. It's not all cell division. There is some entry into quiescence and some cell death occurring, and that gives us a better estimate of what's actually happening within the population. But in the drug-treated conditions, these are actually a lung adenocarcinoma cell line with a mutation in the epidermal growth factor receptor that makes it really sensitive to a particular drug that targets the EGF receptor, that called erlotinib in this case. And in 2 different concentrations of erlotinib, we get very different dynamics of the response with an increasing amount of entry into quiescence and somewhat of an increase in death. This is just an example of the type of characterization we're doing at the cellular level, and we're trying to apply this to breast cancer cells themselves to make the predictions. I don't think I have, that was it, right? Yeah, so in order to better jive to make our measurements match more directly with the in vivo imaging, we've switched over to trying to do joint analyses where we can do both in vitro and in vivo, or microscopy and other imaging-based querying of the same exact cell, so we can couple these types of systems. And I'll throw it back to Tom. Thanks.
Jacob, did you want to ask your question? Jacob, did you want to ask your question?
Yeah. So the simulation here seems, you showed some differential equations before. When you're running the simulation, is it deterministic, or are you playing with parameters? Are you playing with maybe Monte Carlo simulation and have multiple results, either for different parameters or different random seeds? Basically, my question is, do you have only one result, or do you have multiples which you can later on compare to actually what you had in the end?
Yeah, they're all deterministic. The way that we assess the error in the prediction is to do things akin to Monte Carlo simulations. We have estimates of the error in the measurements, and so we can do Monte Carlo simulations to sort out if given initial conditions, we know that initial condition is within plus or minus X% of reality. So we can jimmy that up and then run the simulation again forward and do that 1,000 times to get to get errors on the predictions at the end. So it's a deterministic system, but we can run it through many times just using normal Monte Carlo estimates of noise.
Okay.
Yeah.
Thank you.
Right. Thanks for the question. Right. So in the last few minutes here, this is a system that we've spent some time trying to get up and running. There it is. So this is a bioreactor system, and like Darren said, using this system, we can interrogate it both with microscopy to do what he was talking about, but we can also do MR and PET on it. And right now, this is just sort of the direction that we're going forward. It's a new experimental system that we're pretty excited about. So this is a bioreactor. These things have been around for at least a couple of decades. They're used in the pharmaceutical industry and in infectious disease world a whole lot. This is a cross-section of it. This is just a photograph, and then this is just a cartoon depiction. And so here's the fibers themselves, and you seed cells in between the fibers, and then the nutrients are media is flushed through the bioreactor. Here's the inlet, and here's the outlet, and the media exchanges across the semi-permeable membrane and feeds the cells, and they grow there. And it's MR compatible, and so you can do imaging on it, MR imaging and PET, for example. So here's some examples. This is front view, side view. with two different types of bioreactors we've been playing with. These are with really really densely packed and very small. They have very small interluminal distances. It's about a little less than 100 microns in the top row. In the bottom row, it's about 600 microns across. And so this is diffusion imaging that's been converted into cell number maps using that transformation that I alluded to earlier. It was in the bottom left-hand corner of the slide on diffusion. And so wherever there's blue, there's very few cells there, and wherever there's red, there's a whole lot of cells there. That's the idea. And so this gives you an idea that we can make these nice cross-sectional images, and we can do it in 3D, and then reconstruct the distributions and take out individual time points. So here's some repeat imaging. This was over a weekend here where these were lung. Were these lung lines? This is a lung line that was treated with erlotinib right here on a Friday, and we came back in on a Monday and imaged. And when we compare the proliferation maps that were used from day 14 to 16, you get a lot of red areas indicating positive growth and a few blue areas indicating negative growth. But after the weekend, there was now much less much less proliferation indicated by the red numbers, and a lot more blue boxes indicating more death, negative growth, death that is. So that is a sort of a system that we're using to go forward to try to marry the different scales in a well-controlled experimental setting. Sorry, there's just one last slide here to summarize. So we tried to present 3 imaging-driven models, an increasing level of complexity. First, we worked with the reaction diffusion equation and showed how, even though it was numerically stable and you could get predictions when you went in vivo, the predictions were quite poor, and so this was not a full, complete description of tumor biology, at least in the systems we're working in. Then Jared talked about coupling into the surrounding mechanical properties of the tissue, and we did a little bit better, or actually a lot better. And then now we're trying to couple it through a PDE system that has tumor cell number, the mechanics incorporated into that, and then also glucose metabolism through FDG PET, where FDG PET provides a surrogate of glucose metabolism. So, and we tried to discuss linking the physiological to the cellular scale as well. The end goal of this, right, is that you could, if you really have a theory in place, a system of equations that can do a good job of predicting how a patient's going to respond, then you could sort of give the patient therapy in silico with a whole bunch of different treatment regimens. You could systematically adjust therapies, the order of combination, their dosing, the scheduling, all the rest of it, for as long as you can pay the electric bill to run the computer. And then you could, that would be able to, select an optimal, more rational clinical trial for an appropriate group of patients. And then, of course, it's eminently testable in the preclinical setting. You could imagine stacking the deck where you do this stuff up here, and you have a really good candidate and a really poor candidate. You can test those out in vivo in rats and see if it actually pans out. And then it's translatable because it's all based on imaging data that you can get non-invasively. So these are the people who do most of the work. These are our grad students and postdocs, and there's some staff scientists in here. Jared is in the process of being promoted to junior faculty, so congrats to him. Darren is also a faculty member, and then here are our funding mechanisms. And we always like to thank our patients who participate in the studies, because all of the imaging data on the patients are research studies, and those are not from standard of care studies, so they are coming back through the goodness of their heart to contribute. So we always appreciate that. So thank you very much for your time and attention. And if there's any questions, we'd be happy to field them.
Great. Thank you so much, Tom. I would invite everyone to turn on their webcams and unmute yourselves, and to please pose any questions.
I have one question for the imaging display for the DWI, DCE, and FDG. It looks like the colored map is quite matching with each other. Just wondering how to set up the threshold for highlight the change in that.
I'm not certain I understand the question. Are you talking about how is the dynamic range selected to display?
You have the display the highlight color for the tumor part, right? So you have for DCE-MRI, you have it for the DWI.
I know what you mean now. Yes.
Yeah.
Right. So we did a series of studies to try to figure out what the appropriate way was to segment the tumor based on contrast-enhanced MRI. And the results that matched, so we had a period of time where we had about 24 patients we scanned them within a week of surgery, and so we had an estimate of the imaging-based tumor size, and then we had the specimen from surgery, and we found just empirically by trying a bunch of things that that taking an enhancement threshold of 80% gave us a very, very high correlation between our in vivo tumor size and what they actually saw at surgery. So we used the contrast enhanced image set of stack of images. We look for the voxels that have 80% enhancement, and then so that's what we say is our tumor, and then those voxels through the registration after registration are then just copied over to the other scans. So that's what we've done.
But the thing is that the color map for the FDG or for DWI, and I noticed for the K-trans, they're not exactly matching each other there. Some of them are bigger, like use FDG, PET FDG, that the highlight map seems larger compared with DWI. And also DCE is kind of a little bit larger. So for 3 sets imaging data, you need a consistent with the highlight you're changing there. Do you have a kind of way to set up, because you're going to do longitudinal study, right? So this is for one set on one time point, you have these 3 sets of highlighted tumor part with different methods of measuring there. Then, so the question for that time point, what's the condition you said? I'm set this threshold for to truncate for the K-trans, I'm truncated for the FDG. Then next time you come to next time point, are you going to use the same kind of standard or use kind of different standard?
No, it's the same standard for the MR stuff. It's based on this 80% enhancement threshold, which is what we've found most closely correlated with the size of the tumor at
is it K-trans?
Just looking at the enhancement threshold of if a voxel shows an 80% or more enhancement threshold, we call it as that voxel as being part of the tumor. And when we do that, we get a nice relationship between what we say is the tumor based on the MR and what the surgeon says is the tumor based on surgery. So that's our threshold for picking the tumor on MR at all the time points. It's automated, it's hard coded in. Every time a new patient set comes in, the tumor is extracted if it has 80% or more threshold, because that's what we found has the highest agreement with the size of the tumor at surgery.
So, my understanding right, so you use that way to set up tumor size, then within that tumor size region, then you just pick up whatever you have for the K-trans, the DWI changing that ADC or
right.
the FD, okay.
That's right. The K-trans and the diffusion and all the rest of it is mapped independently of the tumor size. After we map it all, then we go and find the voxels that have 80% enhancement, and those are pulled out. And then we do statistics, or in this case, modeling on the remaining voxels.
Then for the longitudinal study, what do you use for the control for your quantification? I know you're doing quantification in imaging now.
Well, I'm not certain. I don't think I'm communicating very well here, because the voxels are picked out based on this 80% threshold at every time point they come in, right.
For the longitudinal study, so next time patient come back again, if you want to do the measurement, what's the control, quality control or the QC control? You're showing that you're comparing on the same time same base for your quantification, for your FDG or for your DCMI or DW-MRI.
So QA/QC is a. I'm not certain I know what
the quantification. The quantification, imaging quantification. When you come back next time, you're doing the measurement. Those kind of, the changes you see observe with FDG or with ADC or with
right. So the scanners themselves have their own QA/QC, which are done independent of us. But then also we've done a number of repeatability, reproducibility studies on DCE, on diffusion, on a whole bunch of different PET metrics, and so we have an estimate of how much error is in each one of these measurements. And that ties into the question earlier about when we were talking about Monte Carlo simulations to determine how good we are at making predictions. The measurement, or the error we have in our measurements, we have established through repeatability, reproducibility studies. So we know what the error is in our measurement. Is that what you're getting at?
I'm just trying to get on it. When you do the longitudinal study, the second time when you do the measurement, you quantify them. Do you have some kind of baseline to compare with what you've done with the previous measurement, with the previous time point measurement.
Right. Well, also
this kind of issue, people doing longitude studying, they all have this issue. I measure this time and measure last time. Are these two data comparable or not? If I said it's comparable, what basis I have the control for them to compare each other?
Right. So that's, I mean, you're right. So that's quality assurance, quality control stuff, and so that make sure that we do a whole bunch of stuff. We make sure that the scanner hardware is functioning. We do phantoms tests to make sure that our T1s, our T2s, our ADC measurements are all right on the ballpark. We have internal controls for diffusion and for some of the DCE stuff, looking at enhancement in the muscle tissue, for example, to make sure that's within range. But we have test-retest studies, so we know what the range of errors is in each one of these measurements. And so when you say compare from time 1 to time 2, we use the same methods, of course, for each time point, and then they're compared statistically. And in the statistical model, we have terms that account for the variation in the measurement. We've written on this, so it's we've spent a lot of time trying to sort that out. But those are the things that we do.
Okay.
Okay. Are there any other questions, comments to the speakers?
Yeah.
Jim?
I have a comment. I thought, Tom, this is a wonderful presentation for showing.
Jim, can you speak up a little bit?
I'm shouting almost. Can you hear me now? Tom, I thought this was a terrific presentation, and in the sense that it gives people an idea of what modeling can accomplish and how it can be utilized in a very practical fashion. And the particular strength that I loved to see was your presentation of the uncertainty quantification out of the Monte Carlo. I think it's a very good demonstration. So my question is this going to be on the website and available to people?
There's 2 papers that we're hoping to submit in the next month or month and a half. Jared has one of them that is in the final stages, and then there's one other one that is being circulated. So, you can give it to Jim B. If he agrees not to share it with too many people. But I think it'll be released, within a month. We'll let her post it in about a month's time.
All right. No, it'll be just a great example to the world at large, and it doesn't matter. And in fact, you have a nice imaging field, a very practical approach, and our pet people would love to see this.
All right, you've convinced me. Let's Stephanie, we can go ahead and we can release it.
Okay, great.
Oh.
Jim, the original plan was, this is being recorded at this moment, and we will prepare it for posting on YouTube, but we were waiting for Tom to give us the go-ahead after his papers were published.
They're just submitted. We don't have to wait for rejections and resubmissions, just submissions. It'll be fine.
Okay, just you send us a note to let us know, Tom. Okay.
Yes, ma'am.
Any other questions, comments? If not, let's give our speakers virtual applause. Thank you.
Thanks, Gary.
Thank you very much for a great presentation. We look forward to seeing many of you at our annual 2014 Multiscale Modeling Consortium meeting, September 3rd and 4th, and satellite meeting on the 5th. So thanks again for everyone for participating.
All right. Thanks for the opportunity. Take care.